Assalamu alaikum everyone. Welcome to my project presentation. This video is presented by, this is me, Samiha. Thank you for giving me the opportunity to present my research on developing an effective vaccine for hepatitis B. I will briefly walk you through the key points, methods, and my findings. So let's start. My research topic is development of an edible vaccine, Yeast, for producing hepatitis B antigen enhanced by HSP70. My field of interest is immunology and genetic engineering. Before starting, it's very important to look onto the background of today's topic. So, the global impact of hepatitis B virus infection and its significant role in chronic liver disease, cirrhosis, and liver cancer highlight the urgent need for universally applicable vaccine. Current human hepatitis B vaccines are produced using hepatitis B surface antigen, HBsAg, purified from the plasma individuals who carry the hepatitis B virus. So, from the description, we can clearly understand that hepatitis B is a disease and perhaps a very deadly disease related to liver. And the only cure for this is vaccines, which we can create from the individual's plasma who are infected with the hepatitis B virus already. So we should know that why my research is important. The production of human-derived hepatitis B vaccine is limited by the availability of infected human plasma and the necessity of using stringent purification processes to ensure the antigen is free from infectious hepatitis B virus and other potential live agents present in the plasma. So the vaccines which are available in the market, the traditional ones, are not that effective, and it requires stringent purification processes because it's created from human plasma, which can be contaminated sometimes. So any alternatives or solution? There is. The yeast-derived hepatitis B vaccine was proven safe and comparable to the plasma-derived vaccine in producing immunogenic and protective responses against hepatitis B, as shown in both animal and human trials. Developed by the Mark Sharp and Dohem Research Laboratories, the vaccines was licensed for general use in the Federal Republic of Germany in May and in the United States on July 23, 1986. Germany in May, and as the first licensed vaccine produced using recombinant technology, it set a precedent for the development of future vaccines through this method. So, should we delve into my research? So my research question is, can yeast expressed hepatitis B surface antigen with HSP70 co-expression can be developed as an effective vaccine to induce protective immunity against hepatitis B? The methodology that I used is, I used complex methods to test the effectiveness of infusing HSP70 into genetically engineered Saccharomyces cerevisiae yeast along with HSPAg antigen. So why did I choose specifically this strain of the yeast, which is Saccharomyces cerevisiae? Because it was the most relevant yeast strain which was required for my studies. And as we know from the past, there was researchers researching on recombinant DNAs of hepatitis B, and they all worked on these particular strain of yeast. Now, I first did PCR polymerase chain reaction to create as many copies as I need for the specific hepatitis B surface antigen and HSP70 shock protein, and then I did electroporation, which through electric impulses I infused and genetically engineered my strains, and then I did centrifugation, but after a resting period, and through centrifugation I collected my samples and analyzed my data on it, and then in vitro immune response, where I analyzed the cytokinin levels And at last but not the least, storage condition analysis to understand how effective HSP70 would be in my research. So let's know about PCR. Each of the two plasmid samples undergoes a process called polymerase chain reaction, in short PCR, an efficient method for amplifying specific DNA sequences. The reaction mixture includes PCR buffer, Taq polymerase, which catalyzes DNA synthesis, specific primers complementary to the target gene sequences, and nucleotides, which serve as the building blocks of the new DNA strands. And the next step is electroporation. Electroporation is a technique in which a short high voltage electrical pulse is applied to yeast cells suspended in an electrically conductive medium. In this case, I use Sidvia, temporarily permeabilizing the cell membrane to allow plasmid DNA entry. Optimal electroporation conditions involve a high voltage between 2 and 2.5 kilovolts, a capacitance of 25 microfahrenheit, and a precisely controlled pulse duration ranging from 3 to 4.5 milliseconds. Then centrifugation. Randomly selected samples are collected from the yeast culture after 3 days of incubation. These samples undergo centrifugation, which separates the yeast cells from the culture medium, which also concentrating the target cells and proteins for further analysis. Why did I mention 3 days of incubation? Because that was the resting period for those strains after going through such high electric impulses. They needed the time to recover before proceeding to centrifugation. And then in vitro immunoresponse analysis, which is a quite complex method. An in vitro immunoresponse test is carried out to measure the immunogenic potential of proteins produced after the successful completion of the protein expression process. Cytokinin production, a key indicator of immune function, can be quantified using techniques such as enzyme-linked immunoabsorbent assays, ELISA, and enzyme-linked immunospot assays, ELISpot. And finally, storage analysis. Finally, the stability of HBsAg protein in the yeast culture is assessed under various storage conditions. This is critical evaluation helps determine whether large-scale, long-term production of the protein is feasible. The yeast culture is exposed to different temperature conditions over extended periods to observe whether the protein remains stable and any loss or degradation in protein activity is quantitatively measured. So it's results time now. Let's look into the results for my studies. First, centrifugation results. The results showed that yeast cells co-expressing HBsAg and HSP70 produce significantly more HBsAg protein than those expressing only HBs surface antigen. This suggests that heat shock protein 70 enhances HBsAg expression and stability by aiding proper protein folding and preventing aggregation of misfolded proteins. And then immunoresponse analysis, we can clearly see through the chart that the HBsAg plus HSP70 strain is much higher than those our control and empty vector and with only HBsAg strain. Yeast strains co-expressing HBsAg and HSP70 showed higher HBsAg, which is the hepatitis B antigen surface expression, and stronger immune responses with a 30% increase in cytokinin production, IL-2 and TNF-A in dendritic cells, which indicates that it would cause a very fast immune response in human body. The t-test p-value of 0.099413 supports rejecting the null hypothesis, confirming the alternative hypothesis. So the last results of storage condition analysis. Among the batches tested for storage conditions and antigen stability, 50% survived at minus 20 degrees Celsius, 37% at 40 degrees Celsius, and only 13% at room temperature. This suggests that GM vaccine can be stored at minus 20 degrees Celsius, making it a cost-effective option. And we can maintain this minus 20 degrees Celsius through household fridges, which indicates that it's cost effective. Now the conclusion. In conclusion, my studies support the alternative hypothesis, advocating for revolutionary change in immunology through the introduction of GM recombinant edible vaccines. The edible yeast vaccine concept shows promise, particularly for hepatitis B and other infections. Yeast is an ideal expression system for recombinant proteins like HBsAg, which can be incorporated into foods for oral immunization. However, challenges in antigen stability, immune enhancement, and regulatory approval must be addressed in ongoing research and development. Thank you for listening to my presentation patiently.